Hello my dear students. In the last class we have discussed about some of the aspects regarding the kingdom Monera, that is the prokaryotic cells. We have discussed about the bacteria, we have discussed about the types of bacteria based on the arrangement like bacillus, coccus, spirillum, vibrio, like that. In today's class we will discuss about the ultrastructure of a prokaryotic cell, that is a bacterial cell. Since bacteria is the best example for prokaryotes, I will now discuss about the ultrastructure of bacterial cell. Starting with the bacteria is covered by three major membranes. The outermost membrane being glycocalyx. Being the outermost layer of the cell envelope. Then the second layer is cell wall. Just like our plant cells, the bacteria has the cell wall, and the innermost layer is cell membrane. Innermost layer is cell membrane. Here all these three layers together, it is known as a cell envelope. All these together, it is known as a cell envelope. Right. Then inside the cell envelope, there is a fluid matrix. That fluid matrix is known as a cytoplasm. Just like the eukaryotic cell, the prokaryotic cell or the bacterial cell also has the fluid matrix inside the membrane, that is known as the cytoplasm. Inside the cytoplasm we have many of the cellular components. We cannot say all those things as cell organelles. Okay, remember, cell organelles are absent in prokaryotic cell, like a mitochondria, chloroplast, Golgi complex, endoplasmic reticulum, and all other cell organelles are absent in prokaryotic cell. Right? We can just say some of the cell inclusions. It means some of the chemicals, some of the non-membrane bound structures present in the prokaryotic cell. Okay, among them most important one is the nucleoid. Okay, what do we say it is as a nucleoid? It's also known as genophore. It's also known as a prochromosome. It's also known as chromosomal DNA. Right? Means one of the genetic material responsible for transmission of all the characteristics to the next bacteria. Right? Here, what is the type of DNA here? We should understand the type of DNA. Here, DNA is long, circular, and single stranded. Long, fine, means it is very much length, having more length. Then what is the meaning of circular here? Right? Because this becomes the major difference between prokaryotic cell and eukaryotic cell. Okay? When we say eukaryotic DNA, the DNA is linear. DNA is linear in case of eukaryotic cell. DNA is circular in case of a prokaryotic cell. Okay, in case of eukaryotes, DNA is linear. In case of prokaryotes, DNA is circular. Then what does it mean by that? Here, linear means the DNA is double stranded, first of all, and here the two ends of the DNA are free from each other. The two ends of the DNA are free from each other. We can name them as 5' end, 3' end, and 3' end, 5' end. Means the two ends of each strand is free of each other. But in circular DNA, in prokaryotes, the two ends are not free. Whatever may be the length of the DNA, but the ends are ultimately fused together. And the ends of the DNA are ultimately fused together. Hence, it is a circular in case of prokaryotes. But in case of eukaryotes, it is linear, having the free ends on both the sides. Okay, it's a major difference between prokaryotic DNA as well as eukaryotic DNA. Remember the major point here in the study of the cell chapter. Okay, so it is a nucleoid having long circular and single stranded, means it is only one strand is there. In case of eukaryotes, it is a double stranded. It is double stranded, right? It is one of the major thing present in our bacterial cell, right? Then some of the prokaryotes have another set of DNA, okay? That additional DNA is known as plasmid. The additional DNA present in the prokaryotes is known as plasmid. So, you may ask a question. Already it has a DNA. It's called nucleoid, right? Then what is the function of another DNA? What is the function of another DNA? Here, this nucleoid is responsible for only normal activities of the bacteria. For example, transfer of the characteristics, and here expression of the characteristics, or during the cell division, during the reproduction. But whenever the conditions are unfavorable, like high temperature, low temperature, attacking of the host or the pathogen, anything, in that case, this cannot perform all those additional functions. In that case, plasmid has a work. One more thing, this plasmid do not depend upon the nucleoid for replication or duplication. It means for division, this will not depend on the nucleoid. That is why the plasmid is named as extrachromosomal self-replicating DNA or additional DNA. Right? Self-replicating or additional DNA. It means it is helping the bacteria in performing the additional works. Okay, let us see what are those works performed by the plasmid here. Here, plasmid is responsible for many of the functions like root induction. It means whenever the bacteria is a pathogen causing some diseases, in that case, this plasmid helps in the formation of roots in the host plant. If I am depending on somebody else, he is called host, I am called parasite, pathogen. Similarly here, when the bacteria wants to infect on some plants to cause the root induction, In that case, the plasmid helps in the root induction. Such plasmids are called as RI plasmid. Then, in some plants, in some bacteria like Agrobacterium tumefaciens, the plasmid helps in tumor induction. Tumor inducing. Then it is called as a TI plasmid. TI plasmid. Sometimes in some bacteria, the plasmid helps in resistance. Resistance to antibiotics. Then it is called as a R plasmid. Then it is called as a R plasmid. Then sometimes the plasmid helps to kill the E. coli bacteria. The plasmid helps to kill the E. coli bacteria. Then Cole plasmid. So some plasmids helps to kill the E. coli bacteria. In that case, the plasmid is known as a Cole plasmid. Cole plasmid. Like that, the plasmids are named depending on their function. One more thing, just remember, it is an additional DNA. Plasmid is an additional DNA performing additional functions of the bacteria. Okay. Because the basic functions are performed by the nucleoid. All those characteristics, transfer and inheritance property is maintained by the nucleoid chromosomal DNA, but the additional work will be done by the plasmid. Right. So based on this only, we can say the bacteria as plus strain and minus strain. Plus strain and minus strain. You may ask, what is this plus minus? Whenever the plasmid is present, it is called a plus strain. Whenever the plasmid is absent, it is called as a minus strain. It means when the plasmid is present in the bacteria is more capable of performing all these functions. But if the plasmid is absent, in that case, the bacteria cannot function, cannot perform all these additional functions. That is why the plasmid is must for the bacteria to survive under unfavorable conditions, like inside the human body, inside high temperature, inside the very low temperature, on the icebergs, in the hot springs. Like that, in all those unfavorable conditions, the plasmid helps the bacteria to survive. Right? That is the specialty of the bacteria having a plasmid. Again, we discuss about this plasmid nucleoid in second PUC in molecular basis of inheritance as well as in biotechnology. Okay, we will find this concept again in that chapter in second PUC. Right. Then other than that, what are the parts present in a bacteria? Let us see again. Okay, this is okay about the bacteria and the genetic material. So other than this, the bacteria, the prokaryotes, the prokaryotic cell contains the infoldings of the plasma membrane. Here we have seen the innermost layer as a cell membrane or a plasma membrane. But in bacteria, the plasma membrane will make the infoldings, finger-like projections or infoldings. These infoldings are known as mesosomes. These infoldings are known as mesosomes. We can see the difference here. Meso, the middle, soma, body. It means the membranous body is present inside the cell, inside the cytoplasm. Right. We'll see what is that membranous body here. let us discuss a brief of the mitochondria here. In eukaryotes, we have a mitochondria. We have a mitochondria with a outer membrane and a inner membrane. Outer membrane is circular, but the inner membrane has the finger-like projections. Inner membrane has the finger-like projections. These finger-like projections were named as cristae. Known as cristae. Those cristae contain the respiratory enzymes. The finger-like projections are known as cristae. Those cristae contain the respiratory enzymes, and those respiratory enzymes help in the process known as respiration. That is breakdown of the glucose to release energy and some other components in eukaryotic mitochondria. In mitochondria, right? But here in prokaryotes, as I said, as you know earlier, the cell organelles are absent. Cell organelles are absent. Then who will perform the respiration? Who contain the enzymes? That function is performed by the mesosomes. Okay, here the mesosomes contain the respiratory enzymes. Respiratory enzymes. These mesosomes exactly function like a cristae of the mitochondria. They function like a cristae of the mitochondria, and they retain the respiratory enzymes that help in respiration. Along with respiration, the mesosomes also help in binary fission. Binary fission means one bacteria divides equally into two bacteria. That is binary fission. In that process, the mesosomes will also help, because on the inner surface and outer surface of the bacteria, means upper as well as lower, there are the mesosomes. The nucleus attaches to the two mesosomes so that it will divide equally. Both mesosome divides as well as the nucleus also divides into two equal halves. That is why it is called as a binary fission. Bi for two, equal divisions, equal fissions. Okay, this is the main function of the mesosome. First function is to retain the respiratory enzymes for a respiration, and the helping in a binary fission, means for attachment of the nucleoid during a binary fission. Right, this is about a mesosome. Then addition to the mesosomes, what other components are present in the bacteria, prokaryotic cell? That is, we can see there is a large cylinder filamentous structure that we know as a flagella. What it is known as a flagella. Flagella. Right. Here the flagella has three parts. I'll just label by alphabets. Alphabet A, alphabet B, alphabet C. Okay, I'll just label here. Alphabet A is basal body. Alphabet B is hook. Alphabet C is filament. Alphabet C is a filament. Altogether it will make a flagella. Okay, here the flagella is the locomotory organ. Flagella is the locomotory organ. That is the locomotion, movement of the bacteria from one place to another place. Hence it is known as a locomotory organ here. Okay, so it is the development of the plasma membrane outside the cell, outside the prokaryotic cell. The plasma membrane starts evagination, means outward growth, to form the locomotor organ known as flagella. Okay, Again we have a classification of bacteria based on the number of flagella. Based on the number of flagella, we will just write briefly about it. Bacteria based on number of flagella. So here we have first one firstly Eutrichus. Eutrichus, secondly Monotrichus, thirdly Amphitrichus, fourthly Cephalotrichus, fifth Lophotrichus, sixth one is Peritrichus. Look at here, Eutrichus, O for no, Monotrichus, single, Amphi on both the sides, two, Cephalo, head, Lopho, bunch on both the sides, then Peri, around. Means when a bacteria do not have flagella, it is called Eutrichus. When the bacteria has flagella on both the sides, it is called single flagella on one side, it is called as a Monotrichus. Then a bacteria with two flagella on either side, it is called as a Amphitrichus. Let me draw it again for understanding better. So flagella on both the sides. Cephalotrichus means a bacteria with a bunch of flagella on one side of the bacteria. Then Lophotrichus is the bunch of flagella on both the sides. Lastly, Peritrichus is the flagella around the body. These are the types of bacteria based on the number of flagella. Okay, depending on the number, we have several types of bacteria. Better you pause the video and write down these types so that you can go further concept. Right, that's about the flagella and being one locomotory organ. Then other than flagella, the bacteria has few more finger-like projections. It means the slender parts, slender structures, and another level of smaller structures. Another level of smaller structures. These little longer structures, what we can see, they are known as Fimbriae, and these slender smaller parts are called as Pili. Singular pilus. Pili or pilus. The main function of the Fimbriae is movement of the fluid around the body or movement of the water if they are in water. Here the main function of the pili is attachment. Attachment to substratum means to the surface of the land or water, anything. Then attachment to the female partner. Attachment to the female partner during sexual reproduction, that is during the conjugation. That is during conjugation. Hence the pili are most commonly known as sex pili. That is, they help in the attachment of the female as well as male bacteria. For example, if I have two bacteria here, one is plus strand, one is minus strand as male and female. Here the pili of this bacteria, help each other. If it is like this, it will help in the transfer of the genetic material from +10 to -10. In that space there is a transfer of a genetic material. This junction is called as a conjugation bridge. This is actually the process of a conjugation, right? So here these are the other parts. Then other than this, major portion of the cytoplasm is covered by the one of the cell inclusion, which are known as ribosomes. Here ribosomes are in prokaryotes, they are of 70S type and made up of two subunits. One is 50S plus 30S. This becomes 80. Then why is 70? Again the question arises. Means 50S larger subunit, 30S smaller subunit. Imagine the larger subunit, imagine the smaller subunit. It is of 30S, it is of 50S. During attachment, here about 10 subunits is overlapping. 10 subunit is overlapping. That is why out of 50, 30, 80, we are considering only as 70S. Right. So in prokaryotes we have a 70S type of ribosomes, and in eukaryotes we have a 80S type of ribosomes. Okay, we'll discuss in later classes. Just remember the ribosomes in prokaryotic cell are of a 70S type. Then the function of the ribosomes is protein synthesis. Function of the ribosomes is a protein synthesis. Okay. So one more thing, remember, for protein synthesis we have DNA, it's must. Okay, we have ribosomes, and one more thing we need, that is mRNA. Okay, the RNA is present inside the cytoplasm. RNA is present inside the cytoplasm, or the DNA undergoes transcription to form the RNA. Sometimes what happens here is many of the ribosomes attach to a single mRNA. Many ribosomes attach to the single mRNA. It is called as polyribosome. It is known as polyribosomes. Means many ribosomes on a single mRNA. This is formed when there is a need of many proteins of the same type. For example, I have 5 types A, B, C, D, E. I want 10 kilo of A type proteins, A. In that case, to synthesize A type of proteins in large amount, same type, we need polyribosomes. Means which have the same amino acid sequence, and they will synthesize the same type of the proteins. Right. Other than this, in cytoplasm we have many more components, such as reserve food material, such as glycogen in the granules. We have phosphate. Then in some of the bacteria, mainly the cyanobacteria, there are gas vacuoles. Gas vacuoles that help in the floating or buoyancy of the bacteria if it is needed. Like that, many more smaller inclusions are present inside the bacteria or the prokaryotic cell, being a part of our cell, the unit of life. Right. This is a brief introduction about the prokaryotic cell or the bacterial cell. So, if you have any more doubts regarding this bacterial cell, please write a comment so that I can reach you by the solution. Right. I will go to the eukaryotic cell in the next class. Before that, better let us discuss some of the differences between a prokaryotic cell as well as a eukaryotic cell in the next class. Till that, keep learning. Thank you.